Good morning. Myself, Dr. Satish Kavatagimath, Associate Professor, KLE Academy of Higher Education Research, College of Pharmacy, Belgaum. Today, I am I have chosen the topic regarding staining techniques. These will be including many simple gram staining, flagella spore, and different other techniques also. Okay. So by the end of this topic, the learner should be able to know these all things, like what is the theory behind these techniques and the principle of the staining techniques, and what is this chromophore, auxochrome radicals, and how the mechanism of staining techniques will work, and the differentiation between gram positive and gram negative organisms. Okay. So for every technique, there should be some principle on what basis this goes on. Like we all know, initially to brief you about the staining techniques, it's also very much necessary that why we are supposed to perform the staining techniques. What is the purpose of staining technique? Like after obtaining the bacteria from pure culture, it has to be identified. Okay. So as we all know that if it is going to get unstained, so unstained bacteria when seen under a light microscope are not visible. So we are supposed to stain them. The dyes stick to the charges. Either they will be having any positive or negative charges of the bacteria without binding appropriately to the background. There are different techniques also which are only like in negative staining. Whatever the dye we are going to use, it will just stick to the background. Okay, in that particular a separate technique is it. So in others, when you're going to talk about this staining technique, the dye is going to stick to the bacteria without binding appropriate to the background. So for this reason, the bacteria becomes distinguished from the whatever the background or surroundings. On the other hand, what happens? Unstained bacteria will be remaining. No? These are practically transparent and cannot be seen easily through a common microscope. It may be light or compound microscope. On an overall, it can be said that this staining technique mainly depends on three major reasons. I mean, it is going to have three major advantages why we are going to perform. Due to staining, if after the process of staining, these microorganisms can be distinguished from the environment. This is first thing. Second is different types of stainings do help you to bifurcate or to distinguish various type of microorganisms. Third advantage is once if you are properly stained, a microbial cell can be seen under desired magnification. Okay. So we all are these all techniques staining why we are performing is just for the identification of unknown bacterial cultures, as it is one of the major responsibility of microbiologists. Okay. So it may also help you for the classification, the science of classification. Nowadays we all know that it is called as taxonomy. According to this Burgess manual of schematic bacteriology, has been the official international accepted reference for bacterial classification. Okay. So in this staining techniques, as it is told to you, these are very bacteria are very small and cannot be seen through this naked eyes. Proper staining techniques and microscopy are required for this purpose of identification of bacteria. For these reasons, these dyes are used. These dyes do impart colorization to the microbial cell. Microscopes gives an enlarged image of that object or the microbe, and staining techniques differ among microorganisms, and all microorganisms cannot be observed through simple microscopy. So here we are going to deal with the important staining techniques which are used in the microbial examination. Okay. So for the purpose of staining, okay, um, for the purpose of staining, we are supposed to know before going for the procedure, before discussing in detail, it's also very much necessary to know about some common terms which are going to be used as like dyes. Okay, these are chemical substances which are used for coloring. This may be used for general as well biological purpose also. Structurally, these are cyclic benzene or benzoic compounds. There are different types of dyes also available used for imparting color. These are again called as dyes, nothing but stains. These are basically of two types, natural and synthetic. Dyes used for forming a bacterial stain are the ones which we call as synthetic. Dyes are of three types like acid, basic and neutral. Acid dyes have negative charges while basic and neutral have positive or no charge respectively. Chemical nature of the dye, how these are. These are benzene rings which are going to contain chromophore and auxochrome. Okay. Side groups. These chromophore are again further categorized into two types, basic chromophores and acid chromophores. Basic chromophores are of again three types, methylene red, methylene orange, which are going to belong to ozo group, where benzene ring is attached to the side chain nitrogen atoms. The dyes such as safranine belong to azine group, while methylene blue are endamine group type. In this group, benzene rings are attached to each nitrogen atom. In this second one category is acid chromophores. Acid chromophores, the nitro NO2 and quinoid group compounds acts as acid chromophores. Good example of this is nitro like picric acid and all. So to go with there are some other things to be known very much necessary like martins and lakes. Okay, martins and lakes. Martin can be described as one of the metal ion which bonds to the tissue, forms the coordination between the complexes with some dyes. Tannic acid is a good example of this, where these martins are used to produce unwanted dyes, to remove unwanted dyes. So when a mordant is applied during the process of staining, it will be causing some process like which is called as chelation. Okay, form either covalent or coordinate bonds with the dyes. So one of the this EDT is one of the chelating agent which stops the blood clotting. Okay, so further as I discussed about this acid dyes and all. Okay, so how this is process of staining goes on, like staining of the cells and tissues is probably one of the combination of physical and chemical process. Phenomena of absorption, adsorption, capillary and osmosis do play a part in this also. Here we are going to have know what the basic dyes, what is their affinity for acidic tissues and of acidic dyes for basic tissues, which will be giving you some information like whatever the chemical reactions occur and whatever the formation of newer compound will be there. Furthermore, the use of single stain to color the bacteria is commonly known as monochrome staining. Okay, so in the process of simple staining, uh, here what happens basically, to know before going to this, one two things are there just to tell you. Oxidizers are used. This oxidizers can be defined as one of the bleaching agent which when applied to the dyes it oxidizes, like potassium permanganate, oxalic acid and borax percyanide are used as one of the bleaching or oxidizing agents. Solvents like excess of dyes washed with treating the dyed sample with the solvent. Generally, commonly tap water is used as one of the washing agent. However, diluted solutions of ethyl alcohol are commonly used as solvents for the laboratory purposes. So rather, the type of solvent is selected depending to the nature of the dye what you're going to use. If you consider whatever the dye you have taken, it is polar substances, then water is used as a solvent. And the contraindicating, this hydrophobic substances do need non-polar solvents, just 70% alcohol, etc. Different tissues and cells require different types of staining. like many of the different category of stains are there, where you will be observing like crystal violet, eosin, ethidium bromide, fuchsin, hematoxylin, iodine, Manchester brown, malachite green, methyl blue, neutral red, osmium tetroxide, safrani. These are different types of dyes which are used. So when we talk about this simple staining, in this method of staining, a single solution of dye is used to import colorization on the bacterial cell. The prepared smear is flooded with specific type of dye and then again it is washed. This is again followed by drying with the blotting paper and the slide is examined under microscope. Common dyes which are used for staining in this simple method are crystal violet, methylene blue and safranin. Saffron can also be used as a secondary dye provided that when you are going to use in different differential staining, there it is for use as a secondary dye. But here in the simple staining for using of when you will use only one type of saffron can also be used. Simple staining is not effective enough, but it will be going to help you for studying the cellular shape. Okay. Here, as I told you, like you are supposed to select a slide which is free from oil. Otherwise, it won't be. It will be like uneven. So, clean slide should be used, handled by grasping only to the edges to the prevent the decomposition of the grease. Okay. Insert the sterilized dope into the culture, withdraw small quantity, and it should not be a small, when too small or too high. So the smear preparation should be in a proper manner. After that, you are supposed to spread the bacterial growth smear and the thin layer you are supposed to do, not exactly 15 to 30 ml in general. Okay. After that, flame the inoculating loop. Furthermore, dyed the smear, supposed to put 4 to 5 drops of the stain on the smear and pour off the stain and rinse gently with slow running water. The water should not directly fall on the smear what you have prepared to the side, so that the excess amount of stain what you have put, it should get drained off. After that, dry it. Examine the preparation under oil immersion lens using a drop of oil. Okay. So furthermore, this gram staining procedure. So here also you are supposed to see that in gram staining, it's a one of the most useful staining procedure. The staining procedure which can make easy for the identification of differential bacterial staining cells and some of their important parts also. This is called as differential staining. In the simple staining, you are only just observing the cellular shapes. Here it is going to differentiate between the gram positive and gram negative. One of the most important procedure, just as I told you, for the differential staining, which is used to differentiate between two bacterial types of their cell wall depending on the chemistry. Okay, in gram staining. So it is one of the most used differential staining procedure. This method was developed by Christian Graham in the year 1984. This method follows like gram staining procedure. Here the bacterial smear are prepared in a glass slide and thereafter it is going to get heat fixed. Okay. So heat fixing is also done. Second thing, a drop or two of the crystal violet is put on the smear. After 1 to 2 minutes, it's the slide is washed under the tap water to remove the excess amount of stains. The washed slide is subject to few drops of grams iodine and waited for 1 to 2 minutes and thereafter washed with tap water for decolorization. The slide is put with 95% ethanol or acetone. Saffron is added to the slide for 1 to 2 minutes. Just wait, and the smear is mounted with glycerin and observed under microscope. So after that observation, you will be observing the differentiation between the colors, like whatever it is gram positive or gram negative. Okay. So furthermore, the observation will be like here. If it is gram negative, it will be in that pattern, gram